Okay, everyone, switch on your videos. So we will be talking about the sexual reproduction in flowering plants. Basically, why flowering plants? Because flowering, the flower in a plant is the organ of reproduction. It is one of the most important organ that a flower has, and it is very, very important as far as the reproduction is concerned. Now, in this particular chapter, you're going to see all the things associated with the reproduction, sexual reproduction process in the flowering plants, such as the morphology, the structure, the processes, all of that. Okay, now, basically, when we talk about the sexual reproduction, in general, there are three kinds of reproduction that are there. The first kind is the vegetative propagation, wherein the vegetative parts of the plants are used to propagate a new plant. After that, there comes the asexual reproduction, which is uniparental. Uniparental means only one parent is involved in that reproduction. Doesn't involve any gametes or anything of that sort. And the last one is sexual reproduction in flowering plants. So here, both the parents are involved, they make the gametes and thereby make the plants. Now, furthermore, let us talk about the events that take place in the sexual reproduction. So basically, there are three major, you can say, stages that takes place during the sexual reproduction. There's pre-fertilization event, fertilization event, and post-fertilization event. Now, fertilizing, like everything that happens before the fertilization, we call it as pre-fertilization event. All the things that will happen after the fertilization, we'll call it as the post-fertilization event. Now, during the pre-fertilization, what's going to happen? Gametogenesis. First of all, the gametes will be formed and then the gametes will be transferred. That is pollination. So, these two things are going to happen during the pre-fertilization stage. After that, there comes the actual fertilization stage wherein the formation of the zygote is going to take place. Okay, the zygote formation is going to happen during the fertilization stage. And then there comes the post-fertilization event, wherein all those things that would happen after the fertilization has been done, meaning the formation of embryo, the formation of a new plant like seed and stuff, those things are going to happen after the fertilization gets completed. So these are all the various, you can say, stages that it is in a nutshell. We have to study it in depth. But before we move forward, I'd have to teach you about this thing. This is going to always be with you no matter where you go, no matter what you study. The ploidy. Ploidy refers to the sets of chromosome found in any individual. So there are these many types of ploidy. You have haploidy, diploidy, triploidy, and polyploidy. Now, haploidy is in the case of gametes, wherein there is only one set of chromosome. Only one set of chromosome. Diploid is two sets of chromosome. That is represented by 2n. Haploidy is represented by single n. And then comes triploidy. What is triploidy? Three sets of chromosomes are there. Okay. So that is represented as 3n. And the best example is the endosperm. And then when more than 3 sets of chromosomes are present, we call it as the polyploidy. So these are the different sets of chromosomes. These things you people all have to study it one by one properly. I mean, you're going to, encounter these words throughout this chapter. So is this ploidy thing clear to everyone?
Yes.
Any doubts?
No.
Okay. Now talking about this particular pre-fertilization structure and the pre-fertilization events that take place. Now, before all of the things, we have to study the structure of a flower. So in a flower, there are 4 whorls or 4 basic parts, the most important parts of a flower. There's androecium, gynoecium, calyx, and corolla. Now, androecium is the male reproductive whorl. and the gynoecium is the female reproductive whorl. These two are the most important parts of a flower. There comes the calyx and corolla, that are the accessory whorls of a flower. Accessory whorls means that they are not directly involved in the process of reproduction, but are a part of a plant. Now, basically, it's like a big chunk of whorls or parts you have to see. Now, in order for the ease of understanding, for the ease of learning, there is one set of each of these things that we have to study in order to understand it better. So, a single unit of an androecium is called as a stamen. A single unit of a gynoecium is called as the carpel, or you can call it as the pistil. Same way, a single unit of calyx is called as the sepal, and the single unit of corolla is called as the petal. Now, many a times in like you can say plants or flowers such as lily and stuff, the calyx and the corolla, they're not separate. So that is a perianth. So single unit of a perianth is called as a tepal. So now we have, found out the single unit of each of the whorls or each of the parts, so that it becomes easier for us to concentrate and study about one thing. Is it clear, students?
Yes, ma'am.
Any doubts?
No, ma'am.
Okay. Now talking about the stamen, which is the male reproductive part of the flower. It has two parts, that is anther and the filament. Now the anther is a bilobed structure that you can clearly see in the diagram, and it is divided in the middle by a longitudinal curve. Whereas the bottom portion is called as a filament. So filament, it is a long and slender stalk. The proximal end of the filament is attached to the thalamus or to the petal of the flower. That's the case. Now note that the number and the length of the stamen. Now obviously if the flower is big, the stamens can be big. If the flower is very tiny, the stamens will also be tiny. So the number and the length of the stamen, it can vary from one flower to another. Like from flower to flower, it would vary. Okay, and see, here are few of the biological terms that you people should remember. That is, the dorsal position in terms of biology is the back position. The ventral position in terms of biology is the front position. Proximal means near, something that is near. Lateral means side, and distal means far away. So this is something you people must remember in order to learn things better. And this is the various, you can say, positions that you will find biologically speaking. Now talking about the anther, it is a bilobed structure and it is dithecous. Dithecous means that it is made up of two theca or layers are present. Then a longitudinal groove separates the theca, meaning right in the middle it gets divided into two parts because of the presence of the longitudinal groove. Now, when you cut a transverse section of an anther, you can see that the anther from the inside is a tetragonal structure. Tetragonal means that it has four sides present in it. See this thing. See? 1, 2, 3, and 4. Four sides are present. That is why we're calling it as the tetragonal structure. And in each of these four sides, you'll have the microsporangia present. So since four microsporangia are there, two of them located in each of the lobe. So all in all, you can conclude that anther, it is bilobed, it is dithecous, and because of the four microsporangia, it is tetrasporangiate. Tetra means four. Is it clear, students?
Yes, ma'am.
Now the microsporangia is going to develop and it is going to form a pollen sac. Now inside the pollen sac, the pollen grains will be made, and the pollen grains are the male gametes in the case of plants. Talking about the structure of microsporangium. Now when you take a microsporangium and you cut it in between, right? You cut it in between. So the structure that you're going to see is this picture right down there. So the transverse section of a microsporangium, it appears circular in outline and it has 4 wall layers. Starting from the outside, there is the epidermis, then there is the endothecium, then middle layer, and lastly, towards the innermost side is the tapetum. Now see, you can clearly see outermost layer, epidermis, then endothecium, then several middle layers, inside of which there is the tapetum, which is basically the nutritive part responsible for nutrition. So now let us see what are the functions of each of these layers. So the epidermis, the endothecium, and the middle layer, they are responsible for protection. Okay, firstly, they'll be responsible for protection and they're going to help in the dehiscence of anther to release the pollen grains. They're going to help in the dehiscence of anther. Then the last layer, which is the tapetum, it provides nourishment to the developing pollen grains. Nourishment. Now, the cells of the tapetum, they have a dense cytoplasm and sometimes they contain more than one nucleus. Many a times they are binucleated. More nucleus means more function it will do. It will provide nutrition to the developing pollen. This is clear, students? The 4 layers and the functions. Now, inner to the tapetum, like right in the middle, you'll find sporogenous tissue. Now, these sporogenous tissue, they are the homogeneous, like they are made up of homogeneous cells, and they are the ones that will be undergoing the process of meiosis in order to form the male gametes that are the pollen grains. Is it clear? Now, let us study the whole entire process of how a microspore is made. So, microspore formation is called as microsporogenesis, the process of the formation of pollen grains from the pollen mother cell through the process of meiosis. Always remember, in order to form the gametes, always meiosis will be taking place. So the whole entire process of the formation of pollen grains from the pollen mother cell will be called as microsporogenesis. Now, as the anther develops, the cells of the sporogenous tissue, they are going to undergo the process of meiosis to form a microspore tetrad. So, firstly, sporogenous tissue will undergo mitosis, and mitosis is equational division. So, after that, microspore mother cell will be made. When the microspore mother cell will undergo meiosis, then a microspore tetrad will be formed, which will make the pollen grains. Tetrad means 4 cells together, just like this one. This third picture is of the tetrad. You see, you can count the number of cells. Second picture is of a dyad. Dyad means 2 cells or 2 cells in one. Now, inside each microsporangium, there are several thousands of microspores, meaning the pollen grains, and they will get, you can say, released into the environment as the anther will burst. Bursting of anther is called as dehiscence of anther, meaning anther is now full of pollen grain. Now, that's the male gamete. It has to go. So, the anther will burst and all the pollens will be liberated into the atmosphere. That's very important for the dispersal of the pollen grains. Now, talking about the pollen grain, which is the male gamete in the case of plant. So, they are usually spherical in shape, mostly. They can be star shape also, they can be oval also. But majority of the pollen grains are usually spherical in shape. In terms of their diameter, they are 25 to 50 micrometers in diameter. And talking about the layers that they have surrounding them, so they are surrounded by 2 layers, outer layer and inner layer. The outer layer is called as exine and the inner layer is called as intine. Okay, this is the name of the layer. Now, The exine part, it is hard on the outside and it is made up of sporopollenin. It's a chemical which is very, very, very hard. Like nothing can degrade this chemical. Okay. It is one of the most organic material that is one of the most resistant. So the outer layer of the pollen grain is made up of sporopollenin. Now, can someone tell me why? Why not any other material? Why only the most resistant organic material? Why? Hmm? Anyone? Why would this resistant stuff be required? Answer, students, speak in the class.
To protect it from agents, external agents.
For what?
External agents to protect
External
it.
agents. Basically, a tough layer will be present in order to protect it from the harsh environment. Because this is the male gamete, it has to be protected very, very much. So that is why. So exine, it is the hard outer layer and it is made up of sporopollenin. Why? Because sporopollenin can withstand high temperature, acid, alkali, like it cannot be degraded. So the pollen grain will be safe. Now, in the whole entire exine layer, where there is a single place where sporopollenin is not present, we say that part is called as germ pore. Okay, in that part, sporopollenin is not there. Why? So as to facilitate the exchange of gases. Understood? So
Yes.
that gaseous exchange can take place. Now, pollen grains, because of the presence of sporopollenin, which is a very hard substance, it is preserved as fossil. Okay, it can be fossilized. The inner wall is called as the intine, and as compared to the outer wall, it is thin and it is continuous. It's not interrupted anywhere. And the inner wall, the intine, is made up of cellulose and pectin. Now, inside the pollen grain, you see two cells. One is the bigger cell. As you can see in this picture, this part is the bigger cell. And this part is the tiny cell. So, the bigger cell of the pollen grain is called as the vegetative cell or the tube cell. Whereas the smaller one will be called as the generative cell. Now, vegetative cell, it is obviously bigger in size, as you can see over here in this diagram. It is bigger in size. It has a nucleus that is shifted to one area and it has a lot of cytoplasm throughout. So that is your tube cell. Whereas then the germ cell is small, the generative cell is small and floats in the cytoplasm of the vegetative cell. And the nucleus is spindle shaped, but there's like a dense cytoplasm. Clear? And this particular cell only itself is a spindle shaped cell. Now, the one of the most important line that you people have to remember is that in the case of 60% of angiosperms, the pollen grains, they are shed at the two cell stage, meaning at this cell stage, when the pollen grain will mature, they will reach this cell stage, then they will be shed. And in the remaining species, other than the angiosperms, then what happens? Meaning in the rest of the 40%, the generative cell will start dividing and it will form two more cells. Then it will become a 3-cell stage. So in the remaining 40%, the pollen grains will be shed at 3-cell stage. And in the majority, 60%, they will be shed at 2-cell stage. Now, pollen grains of many species, they cause severe allergies. Now, you might have seen students that during the springtime, when the flowers are blooming everywhere, there are many people that they have allergic reactions. They start sneezing even when they are near any flower or any plant. It's because of the pollen allergy that they have. They can, like, it can cause a lot of, long-term respiratory disorders called as chronic respiratory disorders like asthma and bronchitis. Any doubts up until now, students?
Ma'am, could you explain again the 40% and the 60%?
See, what happens is that in 60% of angiosperms, meaning majority of the angiosperms, the pollen grains, they have to be shed in any of the case. So when
Yes.
they are shed at two cell stage. When they have one generative cell and one vegetative cell. At this cell stage, looking like this, they will be shed. This is called as a two cell stage. But the remaining 40% that are not angiosperms, that are different plants, in that case, this particular generative cell will divide into two. So now you have 1, 2, and 3 cells. So now in the rest of the 60%, they will be shed at the 3 cell stage. Clear?
Yes, ma'am. Thank you, ma'am.
Okay, so as I was talking about, that there are many pollen grains which can cause pollen allergy, such as Parthenium. Parthenium is the scientific name of carrot grass. This particular plant, it came to India as a contaminant, meaning while wheat was being imported in India, it just looks like that only. It came to India along with it, and then it grew a lot, and it causes pollen allergy, meaning allergic reaction in many people. Other than this Parthenium, there are Amaranthus, Chenopodium that also cause pollen allergy. Now the thing with pollen is that since they are the gametes, they are extremely rich in nutrients, especially proteins. They're extremely rich in proteins. So many a times there are many people that eat the pollen grains. The pollen grains are used as food supplements in the form of tablets, in the form of syrups. Why? Because it increases the performance. And same way, the pollen consumption increases the performance of athletes and racehorses. As a students, how many of you, who can explain the difference between mitosis and meiosis too? What is the major difference? Yes, tell me, what is the major difference between mitosis and meiosis?
Ma'am, in mitosis, the parent cell will divide into two daughter cells, whereas in meiosis, the parent cell will divide into two daughter cells, which will further divide into four daughter cells.
So, yeah, you are absolutely correct. Meaning at the end of mitosis, we have two daughter cells. At the end of meiosis, we have four daughter cells. But
Yes, ma'am.
the mitotically produced daughter cells, they are, meaning they are exact copies of their parents. If the parents were diploid, the daughter cells are diploid. If the parents are haploid, the daughter cells are haploid. Safrina, are you able to hear me?
Yes, ma'am.
Okay. When we are talking about the meiosis, so in this case, if the parent cell is diploid, so at the end of the meiosis, the daughter cells will be half the number of the chromosome. They will be haploid. This is the major difference, and you people should remember. Even if you don't, try to just skim through, like, check it on internet, but you should know the difference. Now, there's a time period for which the pollen grain is going to be active or useful. And that is called as the pollen viability or the pollen validity. So pollen grains, once they are shed, they have to land on the stigma, meaning of the female plant, before they lose their effectiveness or before they lose their viability. So this is the time period. Viability is the time frame during which the pollen grains are going to be active. Beyond that, useless. So these things, it varies from species to species, and it depends upon the temperature, it depends upon the humidity. So pollen viability in some cereal plants such as the rice, the wheat, it is only 30 minutes after they are released. Only 30 minutes. Imagine after half an hour, they'll be useless. Whereas in the members of the family, like the floral family, such as Rosaceae, Leguminosae, Solanaceae, in all of them, the pollen grains will be viable for months. So they can be used for a long period of time. Whereas for them, the cereal plants, only 30 minutes. Then the last topic amongst the male reproductive part is the cryopreservation. Now, you people might have seen that there are seed banks available, wherein you can find all types of seeds. Same way, there can be pollen bank, wherein you can store the pollen grain. And whenever you'd want to fertilize according to your own need, then you can do so. So such pollen can be used as pollen bank similar to the seed bank. But the question is, how do we store the pollen? So the thing is, cryopreservation is a technique wherein you take a pollen grain and you store it in liquid nitrogen at minus 196 degrees Celsius. They'll be viable for a long period of time. And this is very useful in the crop breeding program. Is it clear? All right. So this was about the male reproductive system. That is the this thing, androecium. Now we'll move on to the gynoecium, which is the female gamete. So the female gametophyte, it is the female reproductive unit, and it can be, like a gynoecium can be single, single pistil is there, we call it as monocarpillary. When more than one pistil is there, we call it as multicarpillary. So now many a times the pistils, meaning the female reproductive part, they can be fused. So that condition is called as syncarpus. The first picture that you see is a syncarpus ovary, wherein all the female reproductive parts have fused. The second picture that you see is the apocarpus. Apo means the pistils, they are free. All the ends are free. So syncarpus kind is found in the plant called as papaver, very important. Apocarpus is found in the plant called as mycelia, very important. Now every pistil or the carpel, it has been made up of three parts: stigma, style and ovum. Now stigma is the landing platform wherever the pollen grains would come and land. The style is this long neck portion that you see that connects the stigma to the ovary. Talking about the ovary, it is the basal bulged part. Okay, bulged means it is swollen. Basal means it is present at the base. Now, inside the ovary, you'll have ovarian cavity, which is also called as locule. Inside the ovarian cavity, there is placenta. And the placenta gives rise to the ovule, which is the megasporangia. This will form the female gamete. If you see here, this is the whole entire thing. Inside the ovary, ovarian locule. Inside the ovarian locule, that's placenta. Placenta gives rise to the ovule. And further, this ovule, when the whole entire fertilization gets completed, the ovule will become the seed. The seed that we have, like how mango has a seed in it. So that seed is a developed ovule. The fruit that we consume is an ovary. The ovary becomes ripened and it forms a fruit. Now, there are many times the number of ovules, meaning the number of seeds in a fruit, will be one, such as wheat, paddy, mango. And when the number of ovules are multiple, such as papaya, watermelon, orchid. Is it clear, students? Any doubts?
No.
All right. Okay, now you people note down, then we'll move forward and talk about the megasporangia and then how the process of female gamete formation takes place. So you people note down. Once you're done, please tell me, I'll slow down.
Ma'am?
Yes, Aneesa, I got the message, dear. You were not able to write the first part of the chapter, right?
Yes, ma'am. In the regular file, yes, ma'am.
The biotechnological applications, right?
Yes, ma'am.
Okay, so before the class ends today, I'll give you a little bit time to take this picture. Okay.
Okay, ma'am. Ma'am, this sexual reproduction in flowering plants, you had already taken a class with me till the pre-fertilization structures and events topic.
Okay,
So
I
I hope,
started. This was your trial class topic.
yes, ma'am.
Okay. Safrina, have I done this with you also?
No.
No. Okay. So you write, Aneesa, you join from wherever you have it written. Okay.
All right, ma'am.
Yes. Don't know. Done, ma'am.
Ma'am, could you please zoom in the image? Thank you.
No, I'm done.
All right, students, are you having any difficulty up until now?
No, no.
No.
Okay.
Mhm.
Done, ma'am.
Yes, both of you are done, students. Shall I scroll down?
Yes.
Okay.
Ma'am, could you please zoom in the image? Thank you, ma'am.
Mm.
Ma'am, could you please scroll up? Done, ma'am.
Done, students? Done till here? Shall I scroll down?
Yes, ma'am.
Done, students?
Yes.
Done, students?
No.
All right. Not done? Okay. All right, students, done? Okay, Aneesa, the part, yeah, okay, whatever part is left, don't worry. Like whenever there will be the next class, you can note it down also, okay? And from where, which part do you have to take the photos from, Aneesa?
Ma'am, I had started writing from Green Revolution. Before that, I don't have anything.
Before that you don't have? Okay.
No.
All right, you take a screenshot. Tell me once you're done, I'll scroll down.
Yes, ma'am. Done, ma'am.